I probably should preface this for everyone that this is about the most ad hoc, random Skype that I think we both have ever had, isn't it? And you've been incredibly gracious to let me have some time with you to talk about various things. So thank you for doing that. So yeah, basically it was just a catch up, really. It's been a long time since we've spoken like this in terms of giving some people in the community an update. So I was hoping if I could ask you a few questions.
Certainly.
And you can just go wild with them, Ron. Whatever comes to your mind, you say. So one of the biggest pieces of news that I know that people are kind of stoked about is the new CRC, the Collaborative Research Centre, with Jonas Bergqvist at, I think it's at Uppsala University. I've probably pronounced that wrong. I don't know if you could just give a little overview of that for people in terms of how that will integrate with Stanford and Harvard and various things.
Well, he has a very nice facility with a lot of high-end equipment, and one of the particular values of using him is that he does a lot of spinal tap on patients, and he's very good at it. And that's a concern with some of the patients with how you do a spinal tap, because sometimes they have a lot of pain with that. And he says that he does it in a good way that doesn't have much pain. In fact, he said the patients would rather have a spinal tap than a standard blood draw in terms of level of pain. So it puts it into perspective. And he does have access to ME/CFS patients. He's done a number of experiments sort of funded by the Open Medicine Foundation looking for antibodies. But I think it would be a good collaboration, especially when we would like to do something with the spinal fluid. And
yes.
So right now, that's sort of his area. We can certainly expand that. And also, it's useful to have a validation sometimes. And when you find something you think is really very important, you want to make sure that it's reproducible in a different laboratory. So that's another place that we will use him, because his facilities are pretty much like what we have.
So really, really top end stuff, essentially.
Yes, and we've been talking about maybe doing some kinds of trials with canarine injected into the spinal fluid.
Okay.
And there's they're doing similar experiments like that with in a clinical trial for a different disease, which I don't think I should talk about because it may be confidential. But things of that type was what we would like him to work on. And we may have him try to validate what we find in the metabolic trap and see if he finds the same thing with his patients. You always worry about different artifacts or equipment malfunctions or all sorts of problems, and you don't want to get led onto the wrong in the wrong direction because of instrumentation problems.
Yeah. I remember you mentioning that before, I think in our last chat or in our update about the issues with, I think it was mass spectrometry at the time.
Yeah, it's the amount of tryptophan and canarine in the cells is not very high, and the other major problem is that there aren't very many cells that actually have the canarine pathway on. Probably about 1%. So that all reduces the signal, and I don't know if he has the state-of-the-art mass spectrometers there that have a high sensitivity or not, but that would be what we would explore, and also tell him what we have concluded of how to do the experiment and see if he concurs or he may decide to try it in a different way.
Yes, that's really interesting actually, because I think a lot of people have, or maybe it's just me, but have associated Jonas with uh, kind of um, immunology and the spinal tap stuff before, just from what he's been talking about at the conferences.
Right.
But that's really interesting actually to know that he may be testing, or it may be a plan to test kynurenine injected into the spinal fluid. I know these ideas all kind of integrate, metabolic, immunological and stuff. But yeah, that's I think that's probably something a lot of people didn't know. So that's really good to know.
Well, it's all focused on trying to understand what's really happening in this disease, and also how we might modulate it in some way or other.
Yes.
What you'd like to have is the primary cause, and of course that's always very difficult to be sure that you're actually on the primary cause, or are you on a sidetrack of some effect of the disease.
Yeah. No, that's really good. Really interesting. I think a lot of people will be very interested in that for sure. That kind of brings me on to just another point, which was the metabolic trap itself. Whether there was any news. I know obviously there's some news that you've just spoken about with Jonas, but whether there's any update to the mass spectrometry problems that you can give out, or
well, we've been looking at the error analysis. What is this? What is the standard error in our measurements? And it's pretty high, and that's making us a little bit concerned. And I think we need to have a more accurate determination. And problems are the sensitivity of the mass spectrometer, the number of cells that have the pathway, and then another problem that we realized is that we have to do an incubation in media. We use Eagles media, that's classic, and we didn't realize at the time, but Eagles media has 25 milligrams per ml tryptophan. And so that dilutes our label.
Yeah, makes sense.
And plasma only has 5 milligrams per ml. So I don't exactly. Eagles media was developed like in the 1950s, so they probably didn't even know how much tryptophan was in the plasma. But it worries us that we're putting in pretty high tryptophan. So we have found some Eagles media without tryptophan.
Okay.
And so we're now using that so that we can control the amount of tryptophan that cells are incubated in. And we can increase the amount of label, percent of the label. And that should give us a big, an increase in signal.
Yeah.
Also, Mike Snyder has let us use his very high-end mass spectrometer. He has actually 3 of them. And so, and the postdoc that's running it is excited about this project, so he will pitch in helping us now, and that's good. That's more workforce. And he's very experienced in it. So now we have 2 very experienced mass spectrometrists people. And, so we carried out that experiment all the way through with it changed in the media. We enriched for the dendritic cells by pulling out the T cells and B cells and not including them. We still use the same number of cells, so that enriches for the antigen-presenting cells that are in the blood sample. And then that went into the higher sensitivity mass spectrometer. So all combined, that increases the signal about 100-fold, I think.
Wow.
That's a lot more, that's going to be a lot more accurate. But we've only run one patient so far under these conditions, but we get the same kind of increase in tryptophan and reduction in kynurenine over healthy control.
Okay,
So,
that's good.
But we have samples already collected from patients, and I think enough that we can run. I think on Friday there were 4 samples ready to go into the mass spectrometer. Julie has set aside the cells are frozen, and that we can pull them out and use them. So hopefully that will go quickly. We're hoping to get quite a few cells in healthy control, a patient and healthy control samples in the next couple of weeks.
Yes.
And that'll be a much more rigorous test of the hypothesis.
Yeah.
One of the considerations and complexities of this is that this trap is presumably a cellular-based process. So it could happen in one cell and not in another. Mm-hmm. And once it's trapped, that cell is trapped. It's also possible, of course, the stem cells that are producing the immune cells could be trapped. And then all cells generated from that stem cell would continue to be trapped. These are complexities of this whole thing that we don't totally understand. And so those are also have to get some explorations of that. We would like to try to start some cell culture experiments, and that may be done by making stem cells and then generating the right types of cells from those stem cells. But that requires an expert in that field. We have to find one, either as a collaboration on this project or recruit somebody to the center to do that.
I think that's a really important note, actually, isn't it? That comes up time and time again, is that so much of this is cutting edge research, and it requires almost new tests to be made, or tests that haven't been run before.
That's correct.
And people in very specialist fields to be brought along onto the team, which isn't a particularly necessarily a quick process.
Yeah, it seems like everything that we do has never been done before, so it's always figuring it out.
Yeah. But no, that's fascinating, and give some clarity.
If we have some cell culture and we can reproduce them, we can actually effectively create the disease, get cells that aren't trapped and get them trapped. That would maybe allow us to set up a sample, a modeling system that would allow us to see how we could get them out of the trap.
Yes.
Because all the ideas that we have currently and the modeling all comes from measurements that were made years ago, and there's always a possibility that something isn't quite correct and all that. So it's always important to check everything.
So that makes a lot of sense. I think that's there's a lot of clarity with that. One thing that gets mentioned, obviously being on the forums, and talking to patients and seeing things that are brought up, one thing that I do know gets a lot of attention is the impedance device. So obviously the fact that healthy cells, cells from healthy controls, or as healthy as controls as one can get, with ME blood, show a certain impedance signal similar to ME cells with ME patient serum. Hopefully I've got that right and my brain fog isn't confused, but I don't know if you can kind of elucidate on how that's going, if there's any progress in terms of filtration, if there's molecules that have been found, or basically just what the progress is on that, really, if there has been any since, because it's such a dramatic thing, isn't it, when to see that healthy patients cells seem to be having a very severe response to ME patients' blood. It's quite a headline thing, really, and gets a lot of people excited. So I don't know if you can talk a little bit about that, if that, the current state.
Well, one thing is that we've written up the current status of it simply because we had a very good statistic of the probability that it's this difference could occur by chance was 1 chance in a billion. So that was sufficient to try to do a publication. And that has been reviewed by an engineer about the circuitry and the design, and it's reviewed by a medical person that does diagnostic testing.
Oh, wow.
So they made some suggestions. We made those corrections. The paper is going to Proceedings of the National Academy of Science. I think it's passed the review. It's now at the editor. But we think it's all good to go, and it will be out soon.
Wow, that's very exciting.
So we have done quite a few experiments trying to figure out what it is. We've done some filtrations, and it's large, but now it appears that it's quite large, and it's possible that it's some kind of particle. And of course, this could be mixed with some small molecules contributing and so forth. It's what we're looking for is the thing that causes the biggest effect.
Yeah.
It could be a particle, and so that's sort of where that is at the moment. Trying to figure out what that particle might be. We might try to purify that particle better, and then break it apart and run it and either do a mass spec analysis of it or some other kind of analysis to see what is in the particle. That might give us a good clue.
Yeah.
But the biggest interest is really to try to see if we can find some drugs that might abolish that signal. It's not necessarily a cure or even a treatment, but it's possible, and it would be worthwhile. What kind of compounds can reduce the signal?
Yeah.
So that's an effort that we will make, and but to do that, we really need to change the circuitry so that we can screen. The amount of blood that we require for this is about 1 drop. So a blood draw can do many assays, but we need independent circuits for that, and there isn't a commercial device that will do that. So we have to design it and build it, and so that's sort of where that's at. And the research associate that was working with me, Raheem, has accepted a job at University of California at Irvine in electrical engineering. So that will allow him to bring in new students to help him work on the project. So it represents a bit of an expansion of the efforts. A lot of stuff will still be run here at Stanford because we collect the patient's blood and stuff. So the device development and other parts of that will happen in Irvine. It's not that far away. It's a short flight. But the biological assays will probably continue to run at Stanford.
Yeah. Okay.
Just to save the cost, because if he does the biological stuff too, then he has to have a setup for collecting the blood and a mobile phlebotomist and someone to take charge of the patients, and it would represent a duplication of effort. Since we have to do that at Stanford anyway.
Yes. I think just maybe just to make clear, and I think you already have made it very clear, but I think it's easy. I kind of get pulled into the thought as well that this whole filtration thing is a lot. It almost comes across a lot more simpler than it actually is. It's actually a very complex thing to find this molecule or molecules or particles. I kind of think of it as a kind of fishing net and then trying to scoop out things, but it really isn't quite like that.
Yeah, right.
It's a bit more complicated.
Yes.
But yeah, I do know people are very interested in that in particular. So that's really good to hear some clarification on that.
So with the blood flow device, we've been doing a lot of modeling of the device to try to help in its design. I've made a new fabrication design.
This is the red
blood cells, right?
Yeah.
And that should maybe give us what we're hoping for is something that gives us a bigger distinction. If this is going to be a diagnostic test, then it has to separate out all of the patients from healthy controls.
Yeah.
And then, of course, we're setting that up so that when we start running other diseases, which we are getting samples for that now, we want to look at the different technologies and how they differentiate between chronic fatigue syndrome and the other diseases. The problem we have is that chronic fatigue syndrome is initiated by a stressor, and having another disease is a stressor. Yeah. So it raises a problem in the sense that if you have MS, which is a pretty bad disease, is it possible that you also have chronic fatigue syndrome? And that's pretty much dismissed by the physicians because they even dismiss chronic fatigue syndrome from a healthy person.
Yes.
Coming down. So when I talk with them, it's kind of, they say, well, no, it's very unlikely they have the two different diseases at the same time. But they're kind of similar in many ways. And certainly the MS patients have fatigue. And they say, well, it's the fatigue caused by the MS.
Yes.
So trying to do this experiment and see how other patients behave is a little complicated.
Yes.
It's not so complicated if MS patients don't show a signal, but it is complicated if they do.
Yeah. No, that makes sense because I think a lot of people know already, but obviously red blood cell deformability isn't something found just in, ME/CFS so far. It's been found in, sepsis or lupus and
exactly
lots of diseases. So so are they're working simultaneously obviously on this device, but are the team looking into ways to possibly, with the data they've got so far, any drug targets, or is this a little bit too preliminary?
We haven't set that up. We want to change the device so that we get a bigger effect on the deformability. So that the new device is smaller. The channels are smaller. It causes the red cells to be stretched out a lot more. And we need to improve the imaging analysis. So because we want to be, the problem is that as the cells move through this channel, they're going to get pretty fast. And if it sort of smears out because of the picture isn't taken fast enough, it makes our accuracy low. So we really have to greatly improve on the imaging, and then we have to make sure that imaging analysis is automated. In the past, it wasn't automated, and it took hours of effort on each sample to get the numbers. We also wanted to decrease the variance, and one way to do that is to have the cell go through multiple constrictions and measure it multiple times, and then take the average. And that may tighten up the distribution and therefore the accuracy.
Yeah. Okay, that makes a lot of sense. Again, it's coming back to this constant theme of being on the razor edge of science again, creating I can't really imagine how complex a device like that is, but yeah, just to be able to do that, it's not an established medical tool, is it? So
No.
it's again kind of creating all of these things for biomarkers, which is fascinating. But yeah, it would be nice if they existed already in some ways. But yeah. One of the other things that is very recent is the board members. So obviously there's been a few new board members. I think there's, hopefully I pronounce their names right. There's Jennifer Frankovich, Daniel Peterson, who's an MD, and I think Jennifer Frankovich is an MD as well. And then she is. And then Michael Snyder, who I think he's obviously a PhD, but I think people will recognize his name somewhat from prior talks. And
he does a lot of big data analysis and looking at other diseases. He does a fair amount with similar kinds of diseases, and he's also interested in chronic fatigue syndrome. And he's thinking about it. He's been coming to our working group meetings, and he shows a lot of interest. And we're using some of his facilities. He's in the same building. That's another advantage. We're on the first floor. He's on the second floor.
Yeah.
And so, we agreed when we moved in that we would share facilities so that we didn't have a lot of duplication.
Yes. That sounds sensible. I don't know how it all works, but that sounds very sensible. I mean, in terms of them joining the scientific advisory board, the reasons for them joining it, is it obviously it's different for each person, but I don't know if you can.
Well, Dan Peterson is a lot of experience with the disease.
Yeah.
He's in Incline Village area, and that's not too far. It's a drive, and you don't have to even fly if you don't want to. So it's useful to have him because of a lot of experience with the disease. And in fact, we've talked about a number of projects that we might do in collaboration. A lot of that is with his samples, and he's been trying lots of things as well. So there's a lot of opportunity there, and also giving his vast experience and making suggestions of what to try.
Yeah, because he's quite a very established name, I would say, in the ME community from an MD perspective.
And we had David Bell, and we can still connect to David Bell by phone, but he's really too frail to travel anymore, so he really can't come to our meetings. We keep him posted, but we don't really give him any work to do.
Yes.
And so he's sort of a replacement for David Bell. But David Bell's not off. I mean, it's just that he really can't help us as much.
That's a shame because David Bell is quite a hero, I think, in many ways.
And very knowledgeable and also experience.
Yeah.
And Jennifer is at Stanford, and picked her because she knows a lot about chronic fatigue syndrome, but she also works on PANS, and it has a lot of similarities, and she's tried lots of things. And it's also my feeling is that we really need to learn from other very closely related other diseases, and it may give us an idea of how other types of experimentation to do, or also possible treatments that, in the case of PANS, I think they discovered it a little bit earlier. And so she's tried a number of treatments that has been successful when the patient hasn't been sick for very long.
Okay.
So it's mostly, and she runs a PANS clinic. So she has lots of patients. We will probably plan to evaluate some of those patients with similar techniques that we've used on ME/CFS and see what the similarities and differences are.
Yes. I guess that's a kind of similar to MS. Perhaps I may be well off base with that, but in terms of looking at the similarities between fatigue, between conditions, and seeing if there's, there could be one central player for fatigue, or, a certain number of similar or the same mechanisms between different diseases.
So we do plan now to do some collecting data on patients with fibromyalgia and chronic Lyme, and but we have to raise money from those communities. We do not want to use money donated by from chronic fatigue syndrome patients and caregivers and so forth to do research on a different disease.
Yes.
So the financial stuff will get compartmentalized. We do have some donations from chronic Lyme, so we will do. We're setting up a collaboration with Bill Robinson, who's a rheumatologist, so that adds another person.
Yeah,
he's an MD, and he gets samples from the East Coast. There are organisms on the West Coast, but they're slightly different.
Yeah.
And so we will be using samples flowing in from the East Coast for chronic Lyme.
Okay.
And see how that compares to the ME/CFS data.
Yeah, that's very interesting. That's really good. Yeah, very interesting. I think it's I think I'm correct in saying that just to talk about funding very, very briefly, that the CRRC, the new research centre, that was fully funded by OMF, by patient donations.
I'm sorry, which one you're talking about?
The with Jonas Bergquist at Uppsala, was that was fully funded by patient donations or OMF?
I've gotten some patient donations from Sweden, and will go to him.
Yeah.
We'll try to get more that will then go to him.
Yeah.
But Linda is really more in charge of all the how much we have and where we should put it, and with the advisory board talking about it.
Yeah. Yeah. No, that makes sense. Okay. I guess this is the last question. I've taken up a lot of your time.
And Mike Snyder. So, although we have shared facilities,
okay, yeah,
we
of course,
we probably should do something together. So, one of the projects we've been discussing is try to develop a wearable device like a watch that collects data from the patient. And we both have quite a bit of experience in that field from different points of view. And the goal is really to have a measurement so that we know when patients have gone to their limit and before they crash. So try to make it so they don't crash.
Yes, which would be incredibly helpful.
Yes.
So is that something that would measure potentially something like lactate or?
So we could go with biochemical. That's the kind of things that we have done. It could also be a very sensitive device measuring heart rate variability. And there probably is a signal in that as well that might tell you that there's a level where there's starting to be a physiological change.
Yeah.
Mike has had some success with that with some other diseases, and predicting before there's a damage has been done.
In terms of, this is just me being really nosy now, but is that in terms of heart rate variability you were talking about then, or just generally smart tech?
Heart rate, and you can do a number of things. You can measure temperature. As you get a change in temperature, it's very subtle, but you have to have a high accuracy for this. So most of the devices are not adequate. The Fitbit, the Apple Watch, those are not adequate. The accuracies are not high enough. So you need a much more complex device that measures high accuracy. We're going to look into the lactate possibility.
Okay.
As another possible way to do this. And that would probably be done from sweat. That's the simplest thing to do. We could probably, there are ways to which you can do continuous blood monitoring, putting a needle, a tiny nano needle into under the skin.
Yeah.
But sweat is also very sensitive. Hasn't been looked at in the patients yet, but we can induce sweat electrically, and so the patient does not have to be physically active to cause the sweating. This can be done from someone who's even in bed.
Yes,
and
because
it's not painful or anything of that type. It's a low voltage that can induce. We've worked out the methods for doing that.
Because I was going to say, not that it is necessarily that relevant, but I know that a lot of ME patients don't seem to sweat,
right?
Like they used to.
Don't exercise well either.
Yes.
The other correlation appears that if you're in very, very good shape, you sweat more. So not sweating is actually a sign that you're not in good physical shape.
Yes.
I don't know how accurate that is, but and we've seen that in our trying to do the sweating. People sweat are very different in the amount of sweat, which is what made us focus on trying to figure out how to electrically stimulate the sweating so that it didn't require activity.
Yes. Yeah. I think I read a paper very recently, actually. It was something about some people can, very healthy, fit people can sweat up to 2 liters a day, which seemed an obscene amount.
An obscene amount, yes. So we have a device that has been being tested out now is to measure the amount of sweat.
Wow.
Because the amount of sweat could very well be a good indicator of physical fitness.
Yes. Yeah. I know for myself, being bedbound, I don't think I've sweated properly in about 5 years.
It's a very good, very likely, yes.
Yeah. Which it has its pros and cons, I guess. But
right.
I'd rather be more healthy and sweat more. Okay, I won't take up hardly any more of your time, I hope. I just wanted to talk about the focus over the next few months. So we've got the London, we've got Millions Missing, and we've got the London conference.
Yes, the London conference, right? And then there's a conference next week at this week, end of this week at NIH.
Oh, okay.
That's I don't know how broadly that's attended by international people. They're certainly welcome. It's only a 2-day conference, so it's a little bit small. I think the participants is around 300.
Okay, and is that for kind of? I mean, that's passed me by, that one. Unfortunately, I don't know anything about that. But is that basically for input, in terms of just gathering people's input for their own studies, or is that?
Well, supposedly they're going to do a little bit of their own study, but I think it's going to be pretty small. I thought they were going to do more, and that's why I was excited about seeing what they were doing. But I think it's a fairly small amount.
Okay. That's interesting. And obviously, you'll be at Invest in ME conference.
Yes, I plan to. Yes.
Speaking again, like as per usual.
Well, hopefully we'll have more data on the metabolic trap.
Yeah, which will be exciting. Okay, I guess that's about it, really, Ron. I should probably leave you be and stop interrupting your Sunday afternoon evening. Thank you for being so gracious.
Oh, you're
And,
welcome.
and Janet as well. I know this was extremely short notice. So yeah, I'm very grateful that you took the time to do it for us.
Well, there is one other area I could just briefly mention.
Yeah,
It's kind of a new area, and that's more from looking at sort of an environmental engineering perspective. And we now have a professor that's retired at Stanford in environmental engineering. And he would like to come and I think join us, just to be part of an activity.
Okay.
And because we're seeing things like mercury toxicity in the patients, about a third. We're also seeing uranium in the patients, and mostly from California. We don't know where that's coming from or the consequence of that. And that person will help us maybe with some of the mold experiments, something we've been wanting to do for some time. There's just a lot of complexity in exactly what to do. So
Yes.
one of the things that we are thinking of looking at is developing DNA probes for all the molds. So if they're in the body somewhere.
Yeah.
And then the other one is looking for a collaboration that can detect the mold toxins. That was something that we wanted to do for the severely ill patients. But that never happened because of the company that was going to do it went out of business. And
Okay.
so we
ideal.
have a replacement for them.
Yeah.
And there are a number of people that are trying to do that, and we don't have to reinvent the wheel if we can find someone that can look for the presence of the toxins in the patients.
Yeah. So just very quickly, I know mercury is quite a big thing in the CFS community, and so is obviously mold. But for mercury, I presume again that this is hair testing, isn't it?
This is all hair testing.
Mm-hmm.
And of course, that we worry about the validity of that. The value of the hair testing is that it's pretty painless on the patients, and the patient anywhere in the world can send us a hair sample, and it doesn't have to get refrigerated. You can put it in an envelope and mail it, right? So the collection is very simple. What we don't know for sure is the validity of the hair. You have to worry about contamination of the hair, because it's out there and getting exposed. So if you have something high, you have to worry about contamination. If there's contamination with mercury, I think that's still a problem. That means a person was exposed to mercury. So that one doesn't bother us so much. But if you look at things like copper and zinc and other metals that might be in the environment, then you have to be careful.
Yeah. Okay. Well, I will leave you be to go and then hopefully enjoy your afternoon.
So we've had some meetings with Eric Johnson about that, and we've actually visited him, and he's a person that can help us.
As in the mold. I don't want to call him the mold guy, but there's
he's had some experience, and we always like to gain the experience from the patients. Sometimes they don't use the right scientific terms. Sometimes they misunderstand some of the scientific background, but you listen to them, and you try to figure out what it is that they're experiencing, and then take them seriously enough that you need to. If you're going to dismiss it, you should dismiss it because you can't find any evidence, scientific evidence and testing that it's there. So until you do that, you have to listen.
Yes.
And there very well could be the mold toxins, the mycotoxins that they produce are unbelievably powerful and very scary. So the aflatoxin is the most mutagenic compound known. And so they're incredibly powerful, and there's a reason that they make these things for their own defense.
Yes.
And they're capable of making very large numbers of things under different circumstances.
Yes.
It's something that I think needs to be explored, and so far I don't see anybody really exploring the molds.
No, I know there was Richie Shoemaker. I don't know much about him, but I know he was one of the mold people, an MD or something like that. But I know from reading Julie Raymer and various people's accounts of mold that it's not a particularly easy thing at all to get rid of.
No, it is not easy to get rid of, and she's another person that we'd like to work with. She's been helpful as well. We just figure out what is the right strategy and what are the right people to do the study.
Yeah,
and it's taken us a while, and it's also a financial problem, right? Everything we do, if we start a new project, where's the money coming from?
Yeah.
And we can't write NIH grants because they won't be awarded until you've actually almost finished the project. Because then they'll know it will work. It has to do with, it isn't the problem with NIH particularly. It has a problem with the fact that the amount of money that they have to fund research is so inadequate compared to the research capacity that is present in this country. And when I've looked at research grants, I would say that probably 90% should be funded, but they're funding at the 10 to 15% level.
Yeah.
And they like to say the grants that don't get funded weren't good grants, but I don't think that's correct. They're all good grants. But the review committee found that we're tasked with finding the best 10, 15%. So it's more of a personal choice from the review committee than it is really hardcore best grants. So that just makes it difficult to get research funding, and good ideas don't get funded very frequently.
No, and it obviously places the kind of emphasis on patient donations and people donating to OMF or various organisations to help fund this research, because again, like you say, NIH haven't stepped up.
We will get the funding at NIH, but I realize it's going to take a very long time because it requires you to have a lot already accomplished. So if we show that the metabolic trap is real, then we can probably get grant funds to work on it. But you've already done most of the work.
Well, exactly.
But then you're sure that in fact when you get a grant, because it will for surely be productive, which is true, right? And so that's one of the problems that they're having because of the really lack of adequate research dollars at the National Institutes of Health overall, I think, is a problem.
It also appears to me from when we've talked before and various things that they're not so much interested in exploratory research. I don't know if that's because of funding. I don't know if that's political or a kind of mixture of both, but they don't seem to be interested in observational kind of studies. It's kind of like you said, it's almost once something's almost kind of proven or at the end stage.
When the funding levels get that low, it makes the review committees very conservative.
Yeah.
So you have an exploratory grant that, hey, this might discover something really important, or then it won't. And here's this hypothesis testing where they have a track record. It's been renewed many times. We know they're going to be productive. We have to give them the money because we can't take the chance of funding something that doesn't work. Yes. And it's the conservativeness of not taking chances or doing something new. And it happens with everybody when you start doing these reviews. So, but it makes research progress slow. You want to take the big leaps.
Yeah.
And are very difficult to get big leaps funded. Anyway, that's why we are focusing on donations, because that can come in fast and move the research faster.
Yes.
In the last round, we put a grant in. We're putting a grant in June. In fact, there'll be 2 grants go in June. We're going to try to every round we'll try to get a grant in.
Yeah.
And then that adds to the funding from the patients. But it has to be something that we know for sure is actually working. So the grant that went in, the last grant went in, is on the mercury, because we know that that's right. It's not a lot of patients, and we'll focus on collecting more patients and studying other aspects of that.
Yeah.
We will do some analysis of looking at the hair and the blood, and then we're also wanting to do cellular level, because it's what is in the cell that makes the difference. How much, like for example, mercury is in the cell makes the difference. And if it doesn't get from the blood to the cell, it doesn't matter that much.
No.
And the hair comes from cells, so it might reflect what's in the cell better than the blood does. So we have to sort all this out, and I think the way to do that is to look at the hair, the cells, and the blood from the same person at the same time.
Yeah.
So that would be the new area that we're going to be exploring.
Yeah, that's really interesting. Mercury is quite something I've looked into quite a lot, and it's kind of fraught with difficulties, especially when you come to trying to get the mercury out in someone who is quite ill to start with. So any better diagnostic methods and anything like that will be really helpful.
Well, the mercury could be some of it coming from old fillings. I have all my old fillings removed.
The whole amalgams.
Yeah, I don't think a lot comes from that, because my mercury is not very high, but it also comes from eating fish. And of course, there are other environmental things that you can get exposed to mercury. I mean, a lot. We used to have mercury thermometers, and we have one patient, I think they're in Europe, that broke a thermometer in his backpack.
Oh, wow.
And took out the most of the mercury, but continued to use the backpack. And he's very high in mercury, and it probably came from that, because he was constantly being exposed to a little bit of mercury. So
it's interesting with the fish as well, because obviously fish contain selenium, and I know that's a compound which that is a trace mineral, which is, I think, quite often found quite low in patients, and I know that it can somewhat combat the oxidant effect of the pro-oxidant effect of mercury.
Well, it's probably that the selenium binds to mercury, and then the mercury is actually taking the selenium out.
Yes.
That's probably good that it's binding. It helps to sequester the mercury. But the problem is the low selenium, because the selenium is used for lots of things. And one of them is the conversion of T4 to T3. And we know that a lot of the patients are low in T3. What we don't know is we don't have a connection between the T3 and the mercury. They're not measured. They're measured independently. So we don't know that the patients that have low T3 have mercury, and we don't know that the patients that have mercury have low T3. That's something that we have to actually try to look at.
I don't know. I think I've mentioned this to you before, probably a little bit too much, but I think I mentioned about measuring cellular T3 levels, because that doesn't seem to. I mean, obviously T3 impacts AMPK and oxygen, and it's a metabolic switch of some sort. So I'd personally love to see that at some point in a study, but it's obviously these cellular measures are pretty difficult to do, from what I understand.
Yes, it's hard to do any of these cellular things because of sensitivity to your methods.
Yeah.
And the methods just, for most things, are not sensitive enough. And that's where we need new technology to increase the sensitivity. And boy, is that hard to get grant funds for.
Yeah.
They're very willing to fund you if you've already developed it.
Yes.
But having an idea of what would increase sensitivity, which would have some risk, it isn't getting funded.
No. It kind of makes the blood measurements we have now seem all the more archaic in many ways.
That's correct.
The simple.
You want to measure one cell.
Yeah, yeah.
And when you do a medical test, you take 10 mils. The difference between one cell content and 10 mils is a tremendous difference in amounts. So that's we would really like to be able to measure it in one cell. We would like to do the metabolic trap in one cell. But there is no method is sensitive enough to do that at the moment that we can find. No. But that's what we like to think about and try to see if we can come up with some idea about how to do that.
Okay.
Anyway, that's our long-term sort of things. I didn't cover everything that we're going to try to do in the future, but a lot of this depends on the amount of funding that we get, and also the problems in the funding is that sometimes we get donors that like to start a project, but you often don't finish the project. The project leads to yet another idea, but it's not quite a new project. Yeah. So how do you sustain some of these efforts, like the metabolic trap? We've got funding to look at that. But now there's many other things that we have to explore in terms of if it looks like it's correct, in terms of, how do we get people out of it? Well, it's still the metabolic trap. It's already funded, so how do you get new funding? It's a problem, which NIH grants are great at, because in fact that's exactly where you have if you establish something like the metabolic trap, then it's easy to get renewals after renewal after renewal. So that's why we have to get NIH grants in when we have some what looks like a potential breakthrough, and then get it supported by NIH, because they will sustain funding.
Yeah. So in the meantime, we just carry on as best we can, and as advocates and people fundraising, and hopefully and NIH will step up to the plate and give us a nice fat grant.
I hope so.
Many fat grants. So, okay, I will leave you there because you will have some evening. Just to say thank you to Janet as well. I know she might be there for helping arrange all of this at ridiculously short notice.
Yes.
And
5 minutes is not bad.
5 minutes, yeah. You've got to seize the day, haven't you? So, hopefully people can see you again soon. I guess for most people that will be Millions Missing first, as opposed to the London conference.
Yeah, well, probably the NIH. I think that's going to be live streamed.
Okay. So hopefully people can tune in. I'm sure OMF will put something out about that.
Yeah.
Okay. Thank you so much.
Yeah, I think that's Thursday and Friday.
Thursday and Friday. Okay.
And a Young Investigators meeting on Wednesday, I believe. I don't know I don't know anything about that. I'm not attending that. I'm not a young investigator.
I'm not saying anything. Okay. Thank you both so much. It's been great to speak to you, and I hope you have a really nice evening. We'll speak to you soon again.
Okay.
All right. Take care.
Get rest.
Yeah, I will do. Yeah, it's a bit late.
I'm sure it is. Yes. All right.
It's fine. All right.
Thanks, Ben.
Yeah. Thanks, Ron. And thanks, Janet. Take care.
Yeah,
Bye.
Bye.